Afternoon. Welcome, Philip, Jelly, Anjali, Babu, Krishtimita, Yash. Anjali, how was your exam, Bacha? Received the B certificate. Qualified for B certificate. बाकी पता नहीं है. So, can you please tell me where were we yesterday on the day before yesterday on Saturday? Which topic we have completed? Anyone? Hmm. Okay. Application of DNA. About growth hormone, right? Hmm? You failed to tell me about growth hormone, remember? Very disappointing. Yes. Anjali? Yep. Chalo. Chalo. Anjali, remember about growth hormone? Anything? मुंबई क्या छोरी बताइए. बताओ बेटा. रिमेम्बर. बताओ. बताओ फिर ग्रोथ हार्मोन क्या होता है. टेल मी व्हाट अबाउट ग्रोथ हार्मोन. बताओ. ब्लैंक. हम्म यही आता है बिजली ग्रोथ हार्मोन में. चलो कोई बात नहीं. रिवाइज कर लेना. So we will continue about genetically modified organism or plants. Okay. In your syllabus, it is genetically modified about crops or plants. Okay. So we will discuss about genetically modified plants. Yes, Jerry, tell me. Endoderm or ectoderm? Endoderm, right? Endoderm forms the internal layers of the organs. For example, kidney, for example, lungs, for example, liver, for example, pancreas, for some example, your gut, for example, your pancreas, for example, your heart, each and every internal organs, brain. Hmm. No, mesoderm. See. Mesoderm is which is the internal organ. See, if you अच्छा चलो देखते हैं. देखो, if this is a part of your liver, suppose no, not this is the liver. To draw a correct diagram also. This is a liver, suppose. Okay. So mesoderm is responsible for the what the what we say the structure of organs. Okay, the structure of organ. Okay, and the endoderm. It is the inside structure of organ. Endo means inside, and meso means middle one. Really clear? Or just wait. Just wait for one day. Okay, I'll or just wait for the lecture for the completion of a lecture. Okay, for technology portion when we'll complete it, we'll then again revise all this general science portion if we have time. then we can see each and every portion of it. Okay, चलो बात करते हैं genetically modified. In the previous lectures, I have defined about genes. Do you all know about genes? Tell me. Previously, I have defined about genes. Are you all familiar with with genes? Tell me. Are you familiar about recombinant DNA? See, recombinant DNA, BT crops, genetically modified crops, all these are very much similar to each other, and they are the same processes. Just just about the minute differences we do in the genetic engineering portion. They are they are said to be different, but as of as if of a general statement, we can say that these all are a similar processes. We are modifying, we are changing, we are just putting some extra effort, some extra genes to change the characteristic properties of a plant, of an organism. Okay, so these are known as genetically modified organisms. The process behind is the same one which is we are performing in recombinant DNA. Tell me, Babu. Okay, tell me which uh, enzyme we use to cut. Which enzyme is said to be the Caesar of DNA of genes DNA of a base pair? Which enzyme? Don't see the copy, please, please, please answer this rapidly. Tell me, bro, Babu, are you there? Which enzyme we use to cut the genes or a base pairs? Yes. Tell me, yes, you. Are you all? हाँ क्रिस्पर इस पर पहुंच जा रहे हो क्रिस्पर आपके यू कोर्स में नहीं था खत्म हो गया क्रिस्पर इस पर एंजाइम की बात मैं कर रहा हूँ विच एंजाइम विच एंजाइम इज यूज टू ज्वाइन द टू अरे कॉपी नहीं खोल रहे हो आप लोग क्या रिविजन क्या रिविजन नहीं हो रहा है क्या कॉपी नहीं है रिविजन नहीं हो रहा है restriction hormone and DNA ligase restriction enzymes for cutting, which is said to be the Caesar of of DNA of genes, and DNA ligase is for joining the two base pairs, and the base pairs define the characteristic properties. Base pairs combine to form a proteins, which shows the DNA properties, the genetic properties. Remember? Chalo. So when we talk about genetically modified plants, when we talk about genetically modified plants, then we'll say something that which one is the first which was genetically modified. Okay, the first one which was genetically modified was flower share plant, tomato. I'm writing it. Just written. It's flower share tomato. फ्लैर, हिंदी में लिख दे रहा हूं इसको, प्रोनाउंसिएशन आपको इजी हो जाए। स्क्वायर टमेटो, ओके, गॉट इट। Was first to be first commercialized genetically modified tomato plant of a tomato. Getting it? Okay. Now see, when we say about tomato, tomato has two basic properties. Not this tomato, but a normal tomato has two basic properties. Which one? Tomato has very thin outer layer and the ripening period, the ripening period, okay, is very short. बहुत जल्दी पक जाता है ये. Yes or no? Okay. Tomato has the property of salivation. Not only tomato, but all the fruits, all the vegetables, and all that thing which has a savory flavor, they have a tendency to make saliva in your mouth. That is why it is easy to get taste of the tomato. Because of it, the saliva in the mouth, the amount of saliva in the mouth increases by eating the tomato. Got it? But the problem is, but the problem was what? That the outer layer was very thin and the ripening period is very short. This process, this property is known as pearnating process. Pearnating property. Pearnating property. Got it? Pearnating property. Due to this property of pearnating, what we do is that we have to make it possible to exist for a longer period of time. For that, biotechnologists, the engineers of biotechnology, right, they made a tomato. They made a tomato which was genetically modified, in which this penetrating property was reduced. Got it? Yes or no? Yes. This was done by the company known as Calagene. Calagene. Okay. This is a California-based company. It's a California-based company. Okay. Got it? This was the first tomato. This was the first tomato. This was the first tomato because of which was commercialized, which was given, which was approved by FDA. FDA is what? Food and Drug Administration of US. Food and Drug Administration. Okay, of US. Due to which this was used publicly. Got it? Yes? Clear? Eh? Clear or not? Yes. The second one was the second genetically modified. Now, if I'm asking genetically modified, are you all capable of answering the genetically modified plants? If you are, if you're asked that define, write a 50 words notes upon genetically modified crops? Can it be possible? I'm just giving the examples. I'm just giving the application part of genetically modified crops. But you are all aware. I think you are all aware of the process behind, basic process, the basic concepts of genetically modified crops. Are you all aware of it? I think so. Via recombinant DNA technology. Genetically modified plants is somewhat the process behind is like the same like recombinant DNA technology, in which the genes which we have to change, the property which we have to change is replaced by some other properties, which which is very which is preferable, which is very much preferable with that particular gene. Suppose Suppose my eyes are black and I want to change this gene that my eyes become red or brown or yellow or blue, anything like Aishwarya Rai, right? Then I would take the brown color, the brown color of eyes of the genes of a brown color of eyes of an Aishwarya Rai and replace it with me, right? By cutting the black color with the brown color. With that, what will happen? With that, I might be have the property of a brown color eyes. The basic difference is this. The basic concept is this. Are you getting it, Salonki? The similar way, if we want to change if we want to change the some properties, right? We have to do it. Got it? But genetically modified crops are very much similar like BT crops. BT means bacterial. The genetic properties are taken of a bacteria, hence known as BT crops. In a case of genetically modified crops, we are modifying the genetic structure. When I'm saying genetic structure, it is what? It is about the base pair. When I'm saying genetically structure, it means what? It means a base pair. We are changing the base pair of any plant so that its property is changed, is modified. Salonki, clear? Yes. Hmm. I'm doing it. नहीं, मैंने बोल दिया, इसलिए मैंने कर लिया है। नहीं, प्रेरणा का मैसेज आया था। आई रिसीव्ड द मैसेज ऑफ फ्रॉम प्रेरणा। तो वो मैं कर रहा हूँ। बिकॉज अनुष्का इज एब्सेंट, आई माइट, आई एम सीइंग इट। ओके, चलो। कोई बात। Clear? Is it clear, Salonki now? Now the second one is golden rice. I think you are all aware of golden rice. Anyone which is not aware of golden rice? I just give an example of golden rice in something. Golden rice. Shubham, do you know about golden rice? Okay. See, golden rice is a type of rice, hmm, type of a grain of a rice, okay, which has the property of beta carotene. Normally, we have seen that in the Indian rice, there's an absence of vitamin A. Vitamin A means beta carotene. Okay, so biotechnologists experimented it, the grain of rice, and then they genetically modified the gene of that rice by treating it with the special gene which has beta carotene, and that special gene was of a maize. Maize has the property of vitamin A, but it has the property of beta carotene in it. So they replaced they induce a new property of beta carotene from a maize to a rice, due to which what will happen, due to which the maize genes was induced in a rice, right, due to which the beta carotene property increased in the rice, due to which what happens, it due to which our rice is now having provitamin A, and also the color is now golden. Okay, now we know that our liver secretes in the process of digestion. I just told you that the liver secretes an enzyme which is useful for changing the provitamin A to vitamin A. Now, this as you all know, that two things I have told you, right? When I'm saying pro means inactive. Whenever the thing is inactive, it is included with pro. Okay, provitamin A. Liver secretes an enzyme which converts this provitamin A to vitamin A. So whenever we eat this golden color rice, right, we have vitamin A increase in our body. This was a second example of BT, of a golden rice, of a genetically modified crops. Various other crops are included, but not no use for you all. Just these type of questions are asked. Okay, and just uh, you have to add on some best example when it is included in the current affairs. Okay, till now only two of it we are seeing it: a flash tomato and the golden rice. Okay, let all, yes or no? Yes. So okay, distribute. Sure. Is it clear to all? Golden rice or not? This is it was asked that which type from which gene we take this beta carotene. Okay, what we have. Okay, what we increased in the property of rice. Got it? So these are all asked. The next one. Next one, which is very important, is RNA interference. This question was asked in 2019 itself, right? And I was teaching it since 2 3 years. So this till then the question was not asked, and the question is asked in year 2019. Got it? So it might be possible to have this topic in your exams. It's a very important topic, and it is very much similar to the genetically modified crops. But a single difference is the the property, okay, which we are stopping it. अभी बताते हैं आपको. I'm just going to tell you about the RNA interference. RNA interference, commonly known as RNAi. Okay. See, until now we are seeing that the genes, which is the common property of a DNA, and DNA is the common property of a chromosomes, and chromosomes are the common property of a nucleus. Yes or no? Is it clear to all? Now is it clear to you? This makes a difference from you and the other candidates. They learn, and you are getting induced to it. Now tell me, is it clear to all? When I'm saying about gene, the gene combined with the base pair, okay, base pair combined to form a gene, gene combined to form a DNA, DNA combined to form a chromosomes, and chromosome combines to form a nucleus. Yes or no? Is it clear to all? Tell me. And when I'm saying base pairs are what? When they are linked together, they are known as protein sequences. Are you getting it or not? Everyone, tell me yes or no. Jili, Babu, Anjali. Yes, okay. When I'm saying they are about RNA interference, then you all are aware of the same process, that DNA is replicated to form a replicated DNA. Yes or no? Known as replication. Then the process of transcription is done to change the DNA to RNA. Then the process of post transcription is done. Then the process of post transcription is done to form rRNA, to form mRNA, and to form which? To form tRNA, transfer RNA. Yes or no? Now after this, we are receiving the protein of the characteristic, the heretical characters. Got it? Now tell me that if I want to stop or to change some specific property of anything, of anyone, then what we have to do? There are two possibilities. Got it? There are two possibilities. First possibility is that we must do what? We must change the property with some other one, or we must stop that particular property to be followed to the other, to be uh, for the formation of a for the formation of a protein sequences, which goes to uh, another generation. ये समझ में आ रहा है आपको? See, क्या हो रहा है? Let me try to tell you that there are two types of characteristic properties. One are effective and one are non-effective. I told you, okay? One are effective and one are non-effective. See, non-effective means that, as I told you in the last class, that about the diseases, some are major one and some are minor one. Remember, Yash, Shubham, Justimita, Babu, Dilip, some are major one and some are minor one. When I'm saying major one, then it means what? That they are effective characteristics, effective properties, which are forwarding to the next generation. And when I'm saying minor one, it means what? They are non-effective. They are not very much effective. They might be passed to the gene, to the next generation or may not. We can change it by by fertilization, by mixing of some another gene. But the major one will forward it, will move from one generation to the another. Yes, do you remember it? I just give you an example, all of you. Yes, that diabetes. Remember, diabetes is a minor one, but if it do not changes with the one or two or three generations, that might then it might be the possibility that it becomes the major characteristic property of a gene. Remember? So in that case, we are seeing to change the earlier we are changing the minor one. Now by RNA interference, we are making the changes in the effective genes also. We are trying to make the change in the effective properties also, which are major properties. We are planning to change. We are working on it, and we had worked on it to change the effective property of it. For example, that the coffee has the property of caffeine in it. Got it? Yes, coffee has the property of caffeine in it. Onion onion has the property that when it is cut it into slices, right, tears come out, and this tear comes out of because of a sulfide gas present in it, and this sulfide gas is what is allyl D sulfide. Not to be learned, just it's just for sake of your knowledge. Okay, in onion it is what, it is allyl or allyl D sulfide. Okay, allyl sulfide, which is which is what, which is responsible gas, responsible chemical, which gives tears in the eyes when it is being cutted, but when it is being sliced. Got it? Cotton. Cotton seeds are toxic in nature. You can't eat it. Yes or no? Can you eat the cotton seeds? No, you can't, because they are toxic in nature. Even they whenever you touch with your hands, you can't just use that hand before washing. Okay, you have to wash because they are toxic in nature, they are poisonous in nature. Okay, when touched with your eyes, they can even destroy your eyes, they can even damage your eyes, the cotton seeds. Got it? These the toxicity of the cotton seed is because of gossypol chemical, GOSIPOL, not to be remembered by you all, just for sake of your knowledge. Gossypol, GOSIPOL. Okay, you don't need to ask, you don't need to copy, you don't need to remember all these terms. Okay, just for the sake of your knowledge. Okay? Now, have you seen the colorful roses? Red, white, yellow, black. Yes? No. Black roses are not very common. But red, white, yellow, pink are very common. Delete? Yes? Yes? But have you seen the blue rose? Blue and black rose? They are not common. Because they are because they are not common. They are not common because they they are modified, they are biotechnically modified. Okay, the red color I just can't pronounce it. Okay, very big name. You don't have to remember, but just see. Uh, the red color, okay, of rose is due to dihydroflavonol, dihydroflavonol reductase. Okay, this is an. I'm using A's. It means what? This is an enzyme. Okay, dihydroflavonol reductase. Dihydroflavonol reductase is an enzyme found in the red roses, which gives the red color of roses. Got it? So you are getting it that the property of onion, property of coffee, property of roses, property of tomato. Got it? Property of uh, the opium seed, right? That opium seeds are what? Opium seeds are very, what we say, they are used in a narcotics, okay, as a drugs, right? Opium, poppy seeds, yes. So these are the effective properties. Are you all aware? Are you getting it? What I'm trying to say is effective properties. Yes. So these are, yes, huh? Major and minor. Similar. Yes, yes, exactly. Right. They are acquired in genetic. Right. Exactly. Acquired means they are non-effective, and genetic means they are effective. Yes, they are very much similar. Okay, clear? Sure. So by this RNA interference, what we are doing now? See the process behind. Okay, see the process behind. I told you that DNA replicated to RNA. Sorry, DNA replicated to DNA, then replicated to RNA, then transcripted to RNA, then post transcription. And post transcription is what? Is rRNA, tRNA, and mRNA. Yes or no? So this is known as post transcription. We are doing what? If in the DNA the effective genes, the effective base pairs are kept silent from the RNA, when these effective genes, these effective properties which are transferred from DNA to RNA and then to the protein sequences. But if we stop this process of formation of protein sequences here, then what will happen? Then the gene which is now very effective is got silent and is known as silent gene. Got it? Just after the transcription phase, when the post transcription is performed, in which the RNA having the effective genes will be transferred to the next generation. Just before transferring, when the formation of protein is performed, just before it, we stop, we cut this process, that not to go to the formation of protein. So what will happen? The gene which has the common property will be converted to silent. Will be silent. Just hold on. Hold on. क्या मिला? क्या मिला? 10 के 2? 10 के 2 कैसे मिल सकते हैं? 10 का 1 होगा। नहीं, 10 के 2 पक्का। अरे रखिए ना महाराज, अरे रखिए, रुकिए रुकिए रुकिए, प्लीज 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 रुकिए, मैं मंगाऊंगा नहीं फिर अगली बार से, मैं मंगाऊंगा नहीं अगली बार से। मुझे लगा 10 का 1 होगा यार। आप मानिए, आप नहीं नहीं, प्लीज 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 राजू प्लीज, पानी मुझे पिलाइए बस। पानी इसको रखिए, उसको रखिए, मैं मंगाऊंगा नहीं फिर अगली बार से। उसको रखिए, मैं इसलिए मंगा लेता हूँ तुरंत, यहाँ कुछ ही करना तुरंत मंगाइए। सॉरी, आई फॉरगॉट टू। सुन लिया ना? ऑल ऑफ यू। कुछ नहीं मैम, आई वाज जस्ट आस्किंग माय मेड, राइट। सुनने में कुछ था ही नहीं, नहीं सुना नहीं कुछ, ठीक है वो ऑन हो गया होगा मुझे लग रहा है। अपने मेट से बात कर रहा था मैं, वो पेन मंगाया था मैंने एंड ही वाज नॉट टेकिंग मनी। मैंने कहा कि रखो पैसे को, चलो खैर। हाँ, तो देववी, हाँ, इज इट क्लियर टू ऑल? टेल मी। We are silencing, uh, we are what, we are performing a process in which we are stopping the process of conversion of RNA to the property of RNA, to the genetic property, to the formation of genetic property, so that we can silence that particular gene, which is very much effective to go from one generation to the another. Clear? Is it clear to all? आ रहा हूं। This is application. Hmm. Is RNA is ribosomal, T is transfer, and is messenger. Because this is a type of RNA, na? This RNA, this RNA is consisting of the base pairs. See, RNA are the single strands, and DNA are double strand. Getting it? These RNA have the base pairs which are combining with each other. Got it? To form DNA again, and this formation of RNA to DNA is known as reverse transcription, right? This formation of from RNA to DNA, not from here but from here, is known as reverse transcription, right? After the transcription, after the post transcription is done, after the performances of post transcription is done, okay, then they are combined together again with the process of reverse transcription to form a DNA, and then that DNA will be transferred from one gene to another, from of one generation to the another. नहीं समझ में आ रहा? कंफ्यूज? कंफ्यूज जिली? See what I'm trying to say. Just see, this is the DNA, Okay, this is the DNA. Got it? It it has a base pair. Got it? But during the mitosis, when it breaks, when the nucleus breaks, got it? In the process of mitosis, what will happen? This nucleus will break. Got it? When this nucleus is breaking itself, then in between this process, स्लाइड में शो ही नहीं कर रहा हूँ ना. Okay, see, when this in the process of mitosis, okay, when the nucleus of a cell breaks, before breaking it, it performs the two process, that these the nucleuses, that the chromosomes, they replicate with each other. Okay, and then after replication they broke down separately. But getting it, this single strain is known as RNA. They then combine together, right? They combine together to form DNA afterwards. And then this DNA will pass to the next generation. Hold on, let me visualize you. Hmm. 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 They are all RNA. Yes, yes, they join together to form DNA. That's what I'm trying to show you. They all combine to form DNA. See, this is process, right? See, mitosis. You don't need to the you don't need to the this is the daughter nuclei, but it from a single nuclei. What happens? See, phases. ये आप लोगों को नहीं पढ़ना है. प्रोफेस मेटाफेस एनाफेस टेलोफेस इसे नहीं पढ़ना है. You say हाँ. See, so this is the replication process. Okay, just before it, what will happen? See, these are the chromatin threads or the chromosomes having in the nucleus. Got it? Okay, this is a nucleus. Huh? They are the chromatin threads of a chromosome. See, these are chromosomes, sister chromatids. Okay, yes, having centrosome in it, and they all have DNA in it. You don't have to bother about centrosome and centroids. Yes. What I'm trying to say is, yes. Now see, yes, this is the process of division. Okay, into a single one, and single one is known as RNA. Okay, Okay, then the two nuclei are formed, and before these two nuclei, see, yes, this is the process. First replication, and then the breaking from DNA to RNA. Now afterwards, they separately join together to form a DNA. Okay, Julie? They first replicate, then they broke down to a single strands having the base pairs, and they then again join together to form a DNA, and then this DNA in the form of a chromosomes is passed to the next generation or the daughter nuclei. Got it? Okay. Yeah. So today we'll have a short class of 1 hour 15 minutes because I'm not feeling well today, and tomorrow we will continue with it. I'll just teach you more 15 minutes, and then we'll close the session. Okay? I'm not feeling well today. Okay. So this silencing of this whole process of silencing us effective genes is known as PTGS. What is this PTGS? This is, let me rub it. What is this PTGS? This was the question asked. Okay, so in 2019, what is this PTGS? PTGS is what? Is the post transcription gene silencing. Okay, the process is what? Post transcription gene silencing, and this whole interference and this whole process in which we are replicating and then moving is the then doing the PTGS process is known as RNA interference. We are interfering. We are breaking the chain from post transcription by doing what? By silencing the genetical property of a gene. This is known as RNA interference. Is it clear to all? Is it clear to all? Tell me all. Yes, no. Acha. See, the whole process of silencing the effective gene by the process of post transcription gene silencing process, PTGS process, is known as RNAi or RNA interference. We are interfering, we are blocking this process of of getting transcripted, of getting post transcripted from RNA to the protein. We are breaking the chain. Got it? Got it, all of you? All of you? Okay. So is it clear to all? Is it clear to all? Now tell me, frankly, we are almost to the end of the biotechnology portion. Only two or three topics are left, which is ICT and which is defense technology. Okay, the major one we are completing it. Tell me, is there anyone which is not comfortable in the class? कुछ समझ में नहीं आ रहा है कुछ not getting it the was not clear in the class the topics which I'm telling कुछ भी समझ में नहीं आ रहा है. Is there anyone? How's the class going? Please tell me. No what? नहीं समझ में आ रहा कुछ नहीं समझ में आ रहा. अंजलि पक्का चलो hope so. चलो फिर topics. After Holi we will do it. Okay. अभी अभी थोड़ा सा रश चल रहा है ना थोड़ा सा क्लासेस का रश चल रहा है सो आई कांट जस्ट गिव यू सम टाइम थोड़ा सा एग्जेशन हो रहा है मेरे पास राइट तो इसके बाद अभी हम होली के बाद वी आर फ्री देन विल डू इट whenever you will get a time just tell me we can do a formal meeting in the through a Google Meet and we can discuss the problems also whosoever have a problem just come to us a common platform and we can have a discussion okay देखो so the applications okay RNAi applications. First, I told you was about blue rose. Okay. Second one, edible cotton seed. We can make. Firstly, it was toxic, but now it is edible. Edible cotton seeds. This is the new type of technology. Okay. Tearless onion. Hmm. Yes, it is toxic. Then you were lucky enough. Then you were lucky. Then you were lucky enough. Go and check your blood. मैं वही कह रहा हूँ ना गो एंड चेक योर ब्लड इट माइट बी पॉसिबल दैट यू आर टॉक्सिक just what I'm trying to say just go and check your blood group it might be possibility that there might be some chances of poison in it बताओ फिर जिली चलो मैं बता रहा हूँ ना it depends on the immunity also it depends on the immunity वही वही चीज है बट इट इज टॉक्सिक यू कैन चेक इट इज टॉक्सिक राइट उसको अभी आपका नहीं मुझे नहीं पता बट इट हैज अ टॉक्सिक इन नेचर यू कांट ईट इट अब वो आप ईट आपने खाया है वो पता नहीं बट यू कांट ईट इट इट इज नॉट एडिबल कॉटन सीड आर नॉट एडिबल राइट सच एज मुझे बताया ना जैसे अभी आप उसको हाथ से आप टच नहीं कर सकते हो मतलब जो लोग करते हैं जो लोग उसको फार्मिंग करते हैं दे आर वेरी मच कंफर्टेबल बट हु आर नॉट दे दे आर नॉट अलाउड टू टच क्योंकि अगर वो टच करके उन्होंने अपने आँख में जैसे सपोज करो मैं बता रहा हूँ ना सपोज करो क्या होता है वन ऑफ माई नीज राइट she drank that good night liquidator. Nothing happened. But we can't say, but we can't say that it is not toxic. Everyone can drink that liquidator. Everyone, one can eat that good night coils. Can we? Can we pause? No. That's what I'm saying now. It is toxic, but it depends on the body to body immunity. We can't have it like a cold drink. चलो आज आज गुड नाइट पीते हैं, आज ऑल आउट पीते हैं हम लोग. ऐसा करते हैं क्या हम लोग? No, we can't. But after the RNAi interference, we can eat the cotton. It has no toxicity in it. Got it? Yes. Then about fourth one. Fourth is decaffeinated coffee. Decaffeinated coffee. Okay. Fifth is opium poppy. Opium poppy. Okay. Which is not harmful. Right. Opium. Got it? Yes. So these are the things. These are the properties. Okay, which we can attain by a process of PTGS, post transcription gene silencing processes, and as the whole is known as RNA interference. Question was asked in 2019. Okay, tomorrow we will try to end this session of biotechnology. Tomorrow we will see about cloning, about DNA fingerprinting, about stem cells. Three topic we have to complete tomorrow, by tomorrow. And by the day after tomorrow we'll see about information communication technology. Okay, so it might be possibility that we have to complete it by tomorrow. Or we whatever we can try. Okay, is it in doubt? No. So for today, we are. I'm sorry. We are. I'm not feeling well. So the class is over. Okay. We meet tomorrow, same time. Okay. Thank you.